we understand that bacteria reproduces asexually. That means that, we talked about binary fission before, and that means that their genetic diversity is not increasing, it's actually decreasing because all the bacteria are too similar to one another. And theoretically, they're all the same, and excluding mutation. But that's not true because bacteria has to find a way to increase its genetic diversity, and it does that through conjugation, transduction, transformation. So transduction is a process that's made possible through the lysogenic phage. And if you remember, when excision happens and the viral genome is now being detached from the host genome, you're going to have some genes that are the host cell's genes being attached onto the viral genes that will then enter the lytic cycle and cause the formation of new progeny that will infect new bacteria. And when this new infection occurs, if the previous host cell genes are also on the viral genes, it's going to cause transduction. So now you're going to have new genes being introduced to the new bacteria, to the new host cell. And then you have transformation. Transformation is when DNA is picked up from the environment. This can be used in labs as well. And the essential of this is that if you have pure DNA, it can be internalized by the bacteria, and it can use its important genetic information to code for new things. And this is another increase of genetic diversity. And the last, we're obviously, this video is about conjugation, is conjugation. So conjugation is a process when we have two kinds, when we have this, it's really dependent on the fertility factor. So the fertility factor is being passed on from one cell to the other cell, or from one bacteria to the next bacteria. And this is the entire concept behind conjugation. So this gives us two kinds of bacteria. A bacteria that has the fertility factor, and a bacteria that does not have the fertility factor. So if you see a bacteria that has the fertility factor, it's a male. And if you see a bacteria that does not have the fertility factor, it's a female. So you might be thinking to yourself, okay, so does this mean that once a bacteria gains the fertility factor, it's converted to a male? Yes, that's the entire concept behind it. And the concept is that all these female bacteria are essentially being converted to males at the end when they receive the fertility factor. So the fertility factor itself is an extrachromosomal element that is passed on to bacteria which do not have it. So we can see the colors here reflecting the gender. Based on this information, what can be determined the goal of conjugation is when it comes to bacterial gender? And we talked about it already, and that is at the end of conjugation, both bacteria have the fertility factor. Now they're the male. So this means that females are being converted to males in the gender sense. And this will allow these male bacteria to interact with their female bacteria and cause more males to be made in a sense. During conjugation, which fertility factor, so the male or the female, has the necessary genes for conjugation bridge to form. Now, obviously, the male will possess these extra genes that are needed to build a conjugation bridge because the conjugation bridge, in a sense, is, if you don't have the extrachromosomal fertility factor, there's no point of having the genes to build a conjugation bridge. So, of course, the male has the genetic information needed to build the conjugation bridge. So the F plus has all the necessary genes to produce sex pili, which contacts the female pili, and once the sex pili and the female pili contact one another, the conjugation bridge is formed. Now, another thing that is important to understand is if we have the female, right, or the male, and we have the female, and this is the conjugation bridge, the direction of the gene transfer is unidirectional, meaning that it's going from one direction and to another destination, and that's the only one way. So this is the extrachromosomal, let's say the fertility factor, and it's being transferred. It's being delivered to the female via the conjugation bridge. So it's one direction. It's important to memorize and understand that. But sometimes we have we have recombination that can happen. And before you get confused, I just want you to understand the only small difference is that you still have conjugation happening. You still have genes being transferred from the male to the female. But the only difference is that when this extrachromosomal element, the fertility factor, sometimes can have recombination with the host cell. So it kind of integrates with the host cell. This genome is now called a high frequency of recombination, or the HFR, which stands for high frequency of recombination. And you're going to understand this in more detail just in a second. But before we do that, let's understand a couple more questions to understand the concept behind this. So let's start with this question. A sample of bacteria includes male and male bacteria. Will conjugation happen? No, it's not going to happen, because remember we just talked that it's unidirectional. It's going, the whole point of conjugation is passing on the fertility factor from the male to the female via the conjugation bridge. And if both of them have it, there's no point of having conjugation. So it's important to understand that there's a specific gender requirement in this entire process. Does the high frequency of recombination considered? Okay, so we just talked about the high frequency, the HFR, and the question is asking us, hopefully you haven't seen the answer already, would you consider this to be a male or a female? And does it have the ability to conjugate? Well, you've already seen the answer now. The HFR still has the genes required for conjugation, so it behaves like a male, so we call it F+. And it has the ability to conjugate as well, as we're going to see in a second. How does recombination occur if bacteria only contain one kind of gene? So you might be thinking to yourself, hey, isn't bacteria doesn't it just have one? How can you have recombination if you just have one kind of genome inside it? And that doesn't really add up, does it? Because the host cell has one chromosome, and we talked about the circular chromosome before. So how does that make sense? Well, that makes sense because as you have, so this is the HFR, and we're going to talk about it in a detailed process 1 second. So look, this is the HFR, the high combination factor, the HFR, let's call that for now, the HFR, right? So it's going to cause it's a recombination factor the fertility factor to replicate. It's going to send it off, and then once this is sent off, you can see that there's some kind of recombination happening over here between the two genes present on both of these, and that is the idea behind recombination. So let's get into this real fast. This is the process through which the entire transfer of the fertility factor happens. So we have the F plasmid over here, which is, the plasmid itself. So remember, the plasmid is essentially responsible for moving these genetic information. These plasmids can be used in genetic engineering as well. So look, we have the male right over here, because it possesses the extrachromosomal element. Now it's going to, then this is the this is the female cell, this is the male cell. Now you're going to see that's going to be passed on to the female. And and this is a copy, by the way, don't be confused. Okay, we don't have anything over here. It's just a copy. It's just showing that we have the extrachromosomal element travel through the conjugation bridge, and it goes all the way to the female. And once inside the female, we have conjugation completed. But remember that this is a copy of the plasmid. There are plasmids over here as well. Then we have the other example, which is the HFR example. So this example, we have the genome, which is integrated. So now it's going to copy off the fertility factor. It's going to send it off. So this is the male, and that's the female. It's going to send it off to the female, and now the female has it as well. And then recombination occurs, and now these both are males officially. They both have the genetic information to cause the fertility, or to cause conjugation. And now a very interesting technique that is used in labs these days is called conjugation mapping. Conjugation mapping is a lab technique that allows the tracking of recombination between so remember we talked about auxotrophs, and auxotrophs are Let me spell it out for you real fast. It's Auxotroph. So auxotrophs are essentially bacteria that are lacking some kind of, let's say, amino acid. It needs a supplement for it to be to replicate. It needs additional help. It's lacking something, maybe caused by a mutation. But if we allow the recombination, let's say this is the bacterial gene, so this is the entire chromosome, excuse me, the bacteria, and that's the bacterial chromosome with the auxotroph, right? And then we have the other gene that comes in, which is essentially the HFR cell. It sends off its fertility factor. Now, when recombination happens over time, and we know that this fertility factor gene has the necessary genes required to fulfill its auxotrophy state, then recombination will happen, and eventually you'll see that, guess what? It's gaining the ability to do or produce for the amino acid it was lacking before, and this is called, and you can track it. Basically, these differences will be tracked on from time. So we'll talk about this in a later video. Hopefully you understood the overall concept behind conjugation.